In this video, we will be discussing selection in Hardy-Weinberg equilibrium as it relates to a population of Care Bears. Now let's get into the basic of Hardy-Weinberg law. In Hardy-Weinberg law, allelic and genotypic frequencies will arrive and remain at equilibrium after one generation of random mating and if all of the following assumptions are met. There has to be an infinitely large population, random mating, no selection, no migration, and no mutation. At equilibrium, the genotypic frequencies are represented by the equation p squared plus 2pq plus q squared is equal to 1, where p is the frequency of the big A allele and q is the frequency of the little a allele. So now we will begin talking about the population of Care Bears in Care Lot, where we have a population of 500 Care Bears. We see 242 pink Care Bears, which are big B, 206 yellow Care Bears, which are big B, and 52 blue Care Bears, which are little b. Now we're going to calculate our allele frequencies. With the information given above, we will calculate the frequency of the big B allele to be 0.69. We will do the same thing for the little b allele and find that frequency to be 0.31. Keep in mind that the allelic frequencies must add up to 1. With those frequencies we just calculated, we can begin to calculate the genotypic frequencies for the population. Taking the equation that we discussed earlier, we will calculate p squared, 2pq, and q squared to be 0.4761, 0.4278, and 0.0961 respectively. We are now going to take a chi-squared analysis of our population. We will use our observed population numbers, allelic frequencies, and genotypic frequencies to find our chi-squared value, which is 0.68202. Since we have 1 degree of freedom in this case, we have a critical value of 3.84 at the 5% significance level. From the chi-squared analysis, we can fail to reject the null hypothesis that our population is in Hardy-Weinberg equilibrium, as 3.84 is significantly larger than 0.68202. Genetically, this helps us to conclude that the B locus in Care Bears follows Hardy-Weinberg proportions. Oh no, a grumpy virus has just been introduced to our population of Care Bears in Care Lot and attacks the yellow and pink Care Bears because of their happiness and cheery attitude. This is an example of selection. As a result of this, the number of pink and yellow Care Bears will decrease, while the number of blue Care Bears will increase. In order to find out how selection affects the population of Care Bears, you will need the fitness of each genotype in the population, and these values are given. After completing the chart, we can compare the initial allelic and genotypic frequencies to those found after selection. Looking at the genotypic frequencies after selection, it is clear that the frequencies of our homozygous dominant and heterozygous genotypes decrease, while the frequency of the homozygous recessive increases. This is what we hypothesized when we found out that the grumpy virus targets are pink and yellow Care Bears. This selection is called directional selection and can be represented by the graph on the right, which shows the initial frequencies in Hardy-Weinberg equilibrium as well as after selection, where there is a clear shift toward our homozygous recessive allele and the blue trait. Scientifically, the fitness that we were given affects genotypic frequencies. If fitness is how well an organism is adapted to their environment, a low fitness would put that specific individual at a disadvantage. Altering the fitness of an individual pulls our population of Care Bears on Care Lot out of Hardy-Weinberg equilibrium.